Hello, good morning, everyone. I hope you had a good weekend. We will finish lecture 2 today. It's probably going to take us 20 to 25 minutes. We just have to do the nucleic acid, and then we are going to start on lecture 3. So I think we didn't have a chance to take up these concept check questions last time. So let's begin with that. First question here. A peptide bond forms between which groups? Now, hopefully, you had a chance to try these things yourself. But if not, that's okay. We can do it now. I'm not gonna launch the poll. I'm just gonna go through it with you to save some time. All right. So you should remember the general structure. I mean, you don't have to draw it, right? But you should recognize what it looks like. So a general structure of an amino acid looks like this. Right? And then I can put another one next to it. And the key thing to remember here is that the side chain, the R, is what distinguish. Oh, you can't see anything? Oh, I'm so sorry. I keep on forgetting that. Oh my goodness. Let me try again. All right. Is it better now? Can you see it now?
Yes, now it's good.
Okay. Yeah. Next time, if you can't see anything, unmute and tell me so I see it right away. Okay. Because sometimes you guys type in the chat, I might not see it. Sorry. Okay. But you guys didn't miss much. Okay. I just drew 2 things down here. The amino acid looks like this, right? And it's the side chain. It's the side chain that makes them different. Okay. The R1, R2 here is what makes up the side chain. Everything else is kind of the same, right, for every amino acid. And if you want to join them together to create a peptide bond, you have to remove an OH from the carboxyl group, and you have to remove an H from the amino group. And then you create the peptide bond right here. All right, so the answer is C. Right? A peptide bond forms between the carboxyl group and the amino group. This is part of the primary structure. You have to remember it, right? Okay. The primary structure is the order of amino acid, right? The sequence of your protein, right? And this is a covalent bond. So covalent bond is not something that can be broken easily, right? You actually have to do hydrolysis to break it, right? The opposite of dehydration synthesis, right? So quite strong to hold the amino acids together. All right, next one. Hemoglobin is an important protein that is composed of 4 polypeptide chains arranged in 3D shapes. What level of protein structure is illustrated by hemoglobin's 4 polypeptide chain? This is one of the questions that is a lot of people get it wrong. Okay, the reason is because I think that when they see the word 3D, they think, oh, tertiary structure, right? And that's what they choose, right? But that's incorrect because here it says the hemoglobin is composed of 4 polypeptide chains, right? So it's 4 things together, right? 4 peptides together to form a protein complex, right? And any protein that has multiple pieces to it, we refer to it as the quaternary structure. All right. I hope that makes sense. If you see this question, make sure you read it carefully. Now, I just want to do a quick recap for you. I already explained to you what primary structure is, right? The sequence that's held together by peptide bond, by covalent bond. Secondary structure is the alpha helix or the zigzag, right? The beta sheets. Those are held together by hydrogen bonds of the backbone. And then tertiary structure is the overall 3D shapes. And there are various interactions between the side chains that holds it together, right? And last but not least, the quaternary structure is when you have multiple polypeptides coming together. Now we can disrupt these structures by simply, cranking up the temperature, right? Or cooling the temperature, changing the salt concentration, changing the pH, or changing the environment that the protein is in, right? All these is in the notes that we did last class, right? So when, let's say you heat it up, right? If you heat it up, you will disrupt the quaternary structure, tertiary structure, and secondary structure. But heat alone is not enough to cut the peptide bond, right? So that you will unravel. I don't have that pipe cleaner that I used last time. But when you heat up, you will unravel it. It will become the original strand, but the peptide bond will still hold the primary structure together. All right, let's take a look at this question. I think we have enough people now, 22 people. You know, let's do a quick poll. All right, just to, you know, wake you guys up a little bit. Go for it. All right, I'm gonna leave it at that. A lot of you are still kind of on the fence for this one, thinking not sure what to choose, and I understand, right? And here, let me explain this situation to you. If people see this, when people see this question on the test, they read the first sentence. They're like, okay, pancreatic cells makes insulin, and they automatically shut down. They're like, what is this thing? What is pancreatic cell? I never heard of it. We didn't do it in lecture. Why is this question on the test, right? And then they get, you know, all confused and stop their thinking process. But the fact is, this first sentence is irrelevant. Okay, like this sentence, you don't even have to understand what it means to do the question, right? Sometimes the application question just puts you in a context, right? Like a scenario, and asks you to answer, right? So here, you don't have to know what pancreatic cell is, and you don't have to know what insulin does, right? What's important is this: the insulin is a type of protein. It's a type of protein. So these cells use something in order to synthesize insulin, which again is a protein. So you can read it like this. These cells use something in order to synthesize protein by the process of something. So how do we make protein, guys? What do we connect together to make proteins?
Enzyme.
I'm sorry, which one?
E.
Okay, so we need amino acids, right? We need amino acids to make protein, right? So that's how you know it's E, and you connect them through dehydration synthesis, right? That's how you answer the question, right? Nucleotides we haven't learned yet, but fatty acid has nothing to do with proteins, right? Saccharides has to do with carbohydrates, nothing to do with proteins, right? Okay, so like sometimes if you see one of these application questions on the test, don't get overwhelmed by it, okay? Just pick out the parts that's relevant to what we've done and solve it that way. I hope that makes sense. Okay, so a lot of people chose B, but that's incorrect. It's like fatty acids and glycerol. That makes like triglyceride, right? Okay, fat. That has nothing to do with protein. I hope I've convinced you why the answer is E. All right, last one here. Let me do the poll as well. I'm gonna go grab something, okay? You guys keep going. Okay, I only have 5 answers. Anyone else before I showed you? All right. So let me just go to the study guide here for a second. I already posted the answers. And so, this question has to do with this chart right here, right? Number 17, right? So the primary structure is the sequence of amino acid held together by peptide bond. Secondary, like I told you, alpha or beta sheets held together by hydrogen bonds. And then tertiary is interaction between the side chains, right? So the question here, primary structure of the protein is determined by the sequence of amino acid. That's correct, right? We're looking for the false one, right? Examples of secondary structure includes alpha and beta sheets. That's correct. We just read it off the chart. Tertiary structure are stabilized by hydrogen bonds of the peptide bone, backbone. And let's see, tertiary structure is interaction between the side chains, right? So that's why this is incorrect. This is incorrect. Side chains, right? A lot of you chose D. Increasing the temperature will disrupt the secondary and tertiary structure, but not the primary structure. I literally just told you this 2 slides ago, right? I said if you increase the temperature, you will unravel the protein, but then the covalent bond, the primary structure, is still going to be intact, right? To break the peptide bond, you need to do hydrolysis, right? Temperature alone is not gonna do it. Do you guys understand? Any questions for me before we move on? Here, I think I remember where my my thing is. Oh, look, this is the protein, right? That I gave you last time. So if you heat up the temperature, it will unravel the protein, and then it will become the primary structure only. But like you can't break this. You can't break this. It's very strong. To break this, you need to like cut it, right? You need to have hydrolysis. So temperature alone, right? Write this down if you didn't get the right answer. Temperature alone cannot disrupt the primary structure. Okay, so that's pretty much it. We're gonna move on to nucleic acid now. Right? This picture here that you see is done using a technique called SKY, all right, SKY. And what it does is it paints the chromosome with different colors. Right? So this technique is useful for diagnosis of certain kind of conditions, right? Because it allows the people to see very clearly if one piece of the chromosome has broken off and reattach itself to another piece. And that actually can happen in some cases, right? So you might have a piece of chromosome 8 that broke off and it attached itself onto chromosome 3, right? It doesn't, you can see it didn't happen here, but if it did, you would see a bit of blue on chromosome 3, right? So it provides a very quick way to visually see if there is any kind of chromosomal disorder, right? So in certain types of leukemia, this is a common occurrence where one chunk of chromosome breaks off and then it goes to another one. All right. So the last macromolecule that we're going to look at is nucleic acid. Chemically, nucleic acid is made up of, the 3 things that are common in all of them, CH and O. And then we also have nitrogen as well as phosphorus. Okay, as well as phosphorus. The monomer for nucleic acid is something called nucleotide. Nucleotide. And there are 2 main types of nucleic acid. Many people already know about DNA. And until the pandemic, people probably have not heard of RNA before, right? But in the pandemic, you'd probably know that some of the vaccines that are available are RNA vaccines, right? So the RNA is the other kind of nucleic acid. Okay, so DNA is deoxyribose nucleic acid, and then the RNA is ribonucleic acid, right? So deoxy means it is missing. missing an oxygen. Okay. I guess we shouldn't say it's missing. It's like, you know, one less oxygen compared to RNA. That's how you can think about it. All right. So let's take a look at the nucleotide itself. Right? The nucleotide has 3 components to it. There is a phosphate group. Okay. Same phosphate group that you will find in what molecule? There is another molecule that we've learned in this lecture that also has a phosphate group to it. Can someone tell me what I'm referring to, please? Your quiz is on Thursday, people. What is the other molecule that has a phosphate group on it?
Are you speaking about ATP?
You are correct, but we haven't learned that yet.
Oh, okay.
Yeah,
Sorry.
yeah. But you're absolutely right. We will learn that later. Another molecule that has a phosphate group on it. I'll draw you a picture, guys, to jog your memory. It looks like this. What molecule is this?
Hydro. Oh,
Okay, so you're right that it has a hydrophobic tail and hydrophilic head.
yeah.
Yes, but what is this molecule?
Phospholipid.
Thank you. Phospholipid. The rest of you know what I'm talking about? I hope so. Thank you, Roy. The phospholipids, yes, that also has phosphate group in it, right? Okay, that's my point. Anyway, it's connected to a 5-carbon ring sugar. Right, so we can represent sugar with like hexagon and pentagon. You should know that by now. And this sugar can be deoxyribose or ribose. Now, I don't know if you noticed, but if a word has a suffix, it feels like English class, right? But if a word ends with OSE, then it's some kind of sugar, okay? Glucose, right? GLUCOSE, right? OSE. Glucose, fructose, sucrose, maltose, galactose, lactose, all these O's, OSE, they are sugar, right? So ribose and deoxyribose are just another kind of sugar. And they have 5 carbons in the ring. And that's represented by our hexagon. Okay, that's the sugar. And it's going to have a nitrogen base attached to it. Okay, nitrogen base. I know it says nitrogenous base here, right? But nitrogen base. It contains nitrogen. All right, so this whole thing is a nucleotide. A nucleotide. All right, now it's very complicated to show you the bond that is formed between the nucleotides. All right, so we're not gonna do that. I'm just gonna briefly show it to you, right? So we have one nucleotide here, one nucleotide here. You're gonna remove an OH from the sugar and an H from the phosphate, and you're gonna form a bond. Okay, so this bond is the covalent bond that holds the nucleotide together, right? Right. They are all held together by covalent bond. Right. The amino acids are held together by covalent bond. We just gave it a special name. We just call it peptide bond, right? But it's a type of covalent bond. And if you forgot what covalent bond is, it's like sharing of electrons, right? The glucose and the glucose are connected by covalent bond. The glycerol and the fatty acid, those are connected by covalent bond as well. Okay. So remember in the previous question where I told you covalent bonds are quite strong, you can't break them just by increasing the temperature. All right, so here is a nucleotide and here's another nucleotide, and you create a bond like this. So that's the actual nucleotide, right? You can see the phosphate here, the sugar here, and then there's the nitrogenous base. Now the one on the left here, this is the DNA nucleotide. Okay, how do I know it's a DNA nucleotide? Well, because if you look at the name of the sugar, it says deoxyribose, right? Deoxyribose. Yeah, I'm gonna explain that right now, okay, Augie? And over here, this is the RNA nucleotide. All right, so take a look at the sugar. The sugar looks exactly the same. The blue sugar and the red sugar, right? The only difference is this location. You see, the ribose sugar has an OH, but the deoxyribose only has an H. So it has one less oxygen, right? That's why it's deoxy. Does that answer your question? Okay, great. All right. Now, the base can come in different versions, okay? For DNA, we have 4 possible bases: A, G, C, and T. A, G, C, and T. All right. Our entire DNA is made up of just 4 letters. It's a bunch of A, G, C's, and T's. Okay. That's the blueprint of life. Right? We're going to learn how to decode it later on in lecture 7. But know that it's only 4 letters. For RNA, instead of T, we have U. Okay. So U substitute for T in RNA. Now they stand for something. Okay, if you learned this before, you probably know what they stand for. But if this is your first time learning it, here are the names. Okay, A is for adenine. Adenine. G is for guanine. That's how you pronounce them. Okay, I know it sounds weird, but that's how you say it. Okay, C is cytosine. T is thymine, which is only found in DNA, and U is uracil, which is only found in RNA. Now, I need you to pay attention to a couple things here, okay? I'll try to write down for you, but it's important that you understand this slide so that you will understand the upcoming slides as well, all right? These are the nitrogenous base, the nitrogen base that you will find on the nucleotide, right? And depending on which one you find on it, they're going to behave differently. Okay, so we can categorize them into 2 groups. Right? We have the purines and the pyrimidines. Purines and pyrimidines. The main difference between the 2 groups is the number of rings in their structure. Purines have 2 rings in it. The way I remember it, because I was really confused when I learned this, when I was younger, right? When I learned this, I keep on confusing the 2. So the way I remember is purine has 2 syllables, right? Like purine, so that's 2 rings, right? Pyrimidines, 1 ring. Okay, so this is 2 rings, 1 ring, and that's going to be important for something else that we will talk about in a bit. What belongs to the group of purines? It's the A, the adenine, and the guanine. And the way I remember it is, you just remember this phrase, pure as gold. Okay, so purines are adenine and guanine. Everything else is pyrimidine. Any question? Do you understand what I'm trying to tell you? No question?
I try to understand.
You try to understand? Okay, but okay, I hope it's not too confusing, but please do let me know, guys, okay? If I can make this any easier for you. All right. Now, just a quick side note, right? There's a condition called gout, G-O-U-T. If you're younger, you probably don't have to worry about it. This is generally something that people, in their after 40s, right? They are more susceptible in getting gout. Gout is a situation where you have a buildup of uric acid in joints, particularly the big toe in some cases, and it causes quite a bit of inflammation. It's a very painful thing to have, okay? So people who have gout have to avoid consuming food that has high purine contents in them, okay? So things like seafood, red meat, beer, that kind of stuff, alcohol, right? They have a lot of purines, right? And if a person who suffers from gout and they eat a lot of these things, it will make the situation worse. It will make the swelling a lot more severe. It's quite uncomfortable. Okay, so you might have heard of that. If not, now you know. All right. let's take a look at our DNA structure here. Okay, think of a DNA as a twisted ladder. That's how most people say it, right? That's a ladder with the rungs. Okay, and then you take the ladder and you twist it, and you get your DNA. Right? It's a double helix. Each side is a strand, and then they loop around themselves to form the double helix. Now take a look at the structure here. Okay, the blue and the yellow, that is the sugar phosphate backbone. Sugar phosphate backbone. All right. So in our nucleotide, right, we have the phosphate and attached to the sugar. I don't know if you remember, I drew that earlier, right, with the hexagon, and then you have the base sticking out, the base, right, and that's going to connect to another phosphate and another sugar, and that's going to connect to, I run out of space, but like another phosphate and then another sugar, right? So the backbone is just alternating sugar, phosphate, sugar, phosphate, sugar, phosphate. That's why it's called the sugar phosphate backbone. And that forms the side of the ladder, okay? The sides of the ladder. The rugs that you climb on, right? Those are the nitrogenous base, right? And in the case of DNA, right, the base could be A, C, T, or G. All right, so I want you to take a look at the diagram here, the picture here. Do you notice some kind of rules in terms of what letters always goes together?
A and T.
Yeah, exactly. Thank you, Maria. So A and T always goes together, and G and C always goes together, right? All right, so I want you to think about this. All right, why does it have to pair in this particular way? Well, what is A? Is A purine or pyrimidine? Okay, try to think using memory. Okay, don't look back. Is A purine or pyrimidine?
Purine.
It's a
Purine.
purine, right? Exactly, because pure as gold, right? I told you, right? So that's purine. And how many rings does a purine have? Two. That's right. Very good, right? So here, A has 2 rings. And thymine is a pyrimidine, which means it only has 1 ring. So the width, right, across the ladder is 3 rings wide. Okay, 3 rings wide. Right, 3 rings wide. Now let's see the G and the C, right? G is also a purine, so it's gonna have 2 rings. C is a pyrimidine, so it has 1 ring, and then that means it's also 3 ring wide across, right? It doesn't matter if the G is on the other side. If the G is on this side, 2 rings, 1 ring, right? So this way, this kind of pairing rule, and this is important, okay? This is the key thing to write down. This ensures the width of the entire ladder is consistent, okay? It ensures the width of the DNA is the same along its entire length, okay? Otherwise, you're gonna get a ladder that looks like this, okay? You're gonna get a ladder that's like sometimes like very wide, sometimes very narrow, right? You understand what I mean? Right? Like if you have an A pair with a G, then it's 4 rings wide, and then sometimes it's 3 rings, sometimes it's 2 rings, and then it's just like bulging and shrinking all over the place, and that creates instability, right? Okay, so we don't want that. That's why we have this pairing rule to ensure the width of the DNA is constant along its entire length. Does anybody have any question about what I just said?
No.
Thank you. Anyone else? Okay, just pop them in the chat, all right? If you want to ask anything. All right? If you don't want everybody to see your question, you can direct message me. That's okay as well. All right, so you guys figured it out already, right? You see that A pair with the T and the G pairs with the C. Which means if you know one side of the ladder, you can actually predict what's on the other side. Okay, so this property is called, we say the DNA strand is complementary. Okay, meaning knowing one strand allows us to predict the other strand. Okay, I'll show you an example in a second. If you take a look at this picture, this diagram, right, you can see the sugar phosphate backbone, right? Sugar, phosphate, sugar, phosphate, sugar. That's the backbone. And sticking out, that's the base pair, right? You see the 2 rings and 1 ring, the purine and the pyrimidine. And there are 2 extra things you should remember from this picture. Number 1, do you see that the strands are upside down compared to each other? Okay, so this property is called anti-parallel. The best way to visualize this is like this. Okay, think of a bunch of people lying on the floor. Okay, so that's like one strand of the DNA, and then you would have another group of people that's also lying right next to the first group, right, side by side, but then they are upside down. What is going on with this guy? Okay, like there we go. Okay, side by side, but upside down, right? So this property is called anti-parallel. Anti-parallel. And then they'd be like holding hands or something. Okay, so that the whole hand holding represents the bond that's holding the 2 strands together. Okay, so take a look here. The A and the T, they have 2 hydrogen bonds. 2 hydrogen bonds holding A and T together. And this here, the C and the G, that has 3 hydrogen bonds. Which means if you have a strand of DNA that has a lot of C's and G's, it would be tighter together. It's harder to pull them apart, okay, compared to a strand that has a lot of A's and T's in it. Okay.
Sorry, I have a question.
Please.
Okay, I didn't understand. Where'd you get hydrogen from? You said 2 hydrogen bonds?
Oh, yeah. So like, okay.
3.
I don't want to go into too much detail, but briefly look at all these the nitrogen base, right? You see there's H here, there's H here, right?
Yeah.
Yeah, so that's where the hydrogen is. But like we don't have to worry exactly how it forms.
Oh.
But hydrogen bond in general is just like just think of it as a glue that holds things together, right?
Oh, okay.
Yeah, so we saw hydrogen bonds between water molecules. We saw hydrogen bonds in proteins holding the secondary, tertiary structure together. And now we have hydrogen bond holding the 2 strands of the DNA together.
Okay, thank you.
All right. So let me repeat, right? If you have DNA that has a lot of C and G's, because they are held together by more hydrogen bonds, it's harder to pull them apart, right? Okay. So again, you're like, oh, who cares? 3 bonds, 2 bonds. Why do I have to know this, right? Can someone tell me what PCR is? What do we use PCR for? I mean, PCR has been around for a long time, okay? But then people start hearing about it during the pandemic. What do we use PCR for?
Is it for the COVID testing?
That's right, COVID testing. You can do an antigen test or you can do a PCR test, right? So PCR test, one step in the PCR test involves separating the DNA strand, okay? And to do that, you need to know how much C's and G's and how much A's and T's are in the sample, okay? Because if it has a lot of C and G's, you need to heat it up higher to separate the strands. If it has a lot of A and T, then you don't need to heat it up as much, right? Okay, so that's like PCR, how it relates to what we're learning here. We will learn more about PCR later on in lecture 9, but just try to connect things for you, right? Because sometimes people feel that there's a disconnect between what we're learning and what happens in the real world. I do try to make the connection for you. Anyways,
Sir?
yes, sorry, you have a question, Maria?
Yeah, what I mean is it's harder to be pulled together.
Okay, so look, right? Here's my props again. So here is the strand, right? One strand, two strand. Remember, what's holding the two strands together, right? Like if I twist them together to make the DNA, what's holding them together is the hydrogen bond between the nitrogen base, right? So if this strand has a lot of G and C together, then they are really tight because the hydrogen bonds between the C and G are 3 hydrogen bonds, right? So they're like, think of it as like 3 pieces of tape holding each of them together, right? So if I want to pull them apart, right, it takes more force to do it. Okay, but if it has only A's and T, a lot of A's and T's in it, right? Only 2 bonds holding together, then that's easier to pull it apart. Does that answer your question, or is still a little bit unclear? Yeah. Okay, great. So, DNA are complementary, as I said, meaning if you know one side, you can predict the other side using base pairing rules. So here we have one strand, right? And we don't have to all unmute and do it. It would be very chaotic. But you can say it out loud, yourself. All right. So what does A pair with? Hopefully, you remember A goes with T, right? And then it's C, C, G. Okay, I hope you're doing it with me. You can hear you do it through the computer screen. T, T, A, T, G, C, C. Okay, so just using the base pairing rule that A always goes with T and C always goes with G, we can predict the other side. Okay, it's not even a prediction. Okay, we know exactly what's on the other side.
Quick question.
Yes, please.
What happens if like the A and C don't, or sorry, the G and C don't go together? What happens if they like mix match?
Yep. That is lecture 7. All right. So hold that thought. Come back and I'll tell you. But short answer is you have a mutation. And if you have a mutation, the instruction to make the protein is going to be different now. You're going to make a protein that's misfolded, and that could affect its function. Like sickle cell anemia that we talked about, right? That's the short answer. All right, so let's take a look at this one, right? This one has a lot of A's and T's, right? A's and T's in it, right? I mean, you have some C and G's, but majority is A and T, right? So this strand would be relatively easy to pull apart. Okay, as opposed to if I have something that's like G, G, C, C, G, C, C, A, T, C, C, G, C, A, T, something like that, right? That's a lot of C and G's. So if I want to pull this apart, it's harder because there's more hydrogen bond holding it together, right? Okay, so 3 hydrogen bond for each of the C and G's, right, making it stronger. Any additional question? Is an example of what? Zena? What do you mean? Oh, mutation. Oh, yeah. Okay, I see. Yeah. I mean, there are tons of things that has mutational basis to it, right? And we're gonna explore more about that later. Okay, this question. Okay, you will see this question probably on the quiz. Let's just try to do it a little bit. All right. It's a bit of math. Okay, but don't worry. It's not too complicated. Suppose the cell has 40% adenine. Find the percentage of T, C, and G. Okay, so 40% adenine. Right away, you should know if there is 40% adenine, something else must also be 40%. What is the other base that is also 40%? Help me out here. Yeah, T, right? Exactly, because A pairs with T. So if you have 40% A, you will also have 40% T. Okay, they are the same. Now together, the A's and the T's would add up to 80%, right? 80%. Which means everything is 100% and you subtract it to 80%, you will have 20% left, right? And Sarah, you are absolutely correct. 20% for the C and the G, and you split them evenly. So divide by 2, you will have 10% C, which equals to 10% G. That's all. Okay. Fairly straightforward. If you need to rewatch this, you will look at the recording, and it is at the 40-minute mark of the recording. Okay, so you want to skip to 40-minute mark. All right, and then just one last slide for RNA. RNA, it's single-stranded compared to DNA, which is double-stranded. The backbone is made of ribose sugar and phosphate, as opposed to deoxyribose. And instead of thymine, we have something called uracil. All right. There are different kinds of RNA. They have different functions, which we will learn later on in the course. But just know that there are different ones. Like there's mRNA, which is the one found in the vaccines, rRNA, tRNA, right? Just different kinds. All right, that's it. That's the whole lecture 2. I know it's like a lot, right? Like I think we have like over 100 slides. Okay, very, very a lot of information. So make sure you study all of that for your quiz. Let's do some poll here. Okay, poll questions. Okay, now please participate. All right, that's the only way for me to gauge if you guys understand or not. When do you get the study guide? It's already online, Medicine. What do you mean? You mean the answers, or do you mean like the actual study guide?
Sorry, the study guide for the quiz?
Oh, the study guide for the quiz is just the regular study guide. Like I don't have like an extra study guide for the.
Oh, okay.
Yeah,
Okay.
sorry.
Okay.
All right, I have 4 answers only. Come on, you can do it. I'm very pushy. Go, go, go. Okay. Thank you for those of you who have attempted. Yeah, the correct answer is the last one. Purine have double rings, right? Double ring the structure. Let's take a look at why the other ones are wrong. Adenine's bonding to thymine is stronger. Nope, it's weaker, right? Than C and G. C and G has 3 bonds, A and T 2 bonds. Cytosine is a purine. Nope, right? It's pyrimidine, pure as gold, right? Remember? Purine binds readily to deoxyribose, but not ribose. So let's take an example of purine, right? Which is A. A is found in both DNA and RNA, right? So it will bind to both deoxyribose as well as ribose. Right? So that's why this is wrong. Both adenine and thymines are purine, and again, that's not true, right? This is pyrimidine, right? So answer is E. Most of you who answered got it right. All right, this is a long one as well, and it's a multi-select question. There are more than one correct answer. Please take a look.
Sorry, Professor. Question B, B there.
Mhm.
I can't see what the last part says. It says adenine.
Adenine forms a base pair with uracil.
Oh, okay. Thank you.
Yeah. Are you not able to move the poll window away? I'm curious.
Yeah, my computer, I'm just having trouble with it.
Oh, okay, sorry. Okay, anyone else? Some of these questions are going to be on your quiz, all right? So make sure you pay attention to them, review them, and yeah. That's all I can say. I don't know what else to tell you. All right, we're looking for the ones that are not true, okay? Not a feature. A nucleotide with a single strand are connected by covalent bond. That is not a feature of DNA because DNA is double-stranded, not single. B, adenine forms base pair with uracil. That is incorrect. There are no uracil in DNA, right? Okay, only thymine. Complementary strands are held together by hydrogen bond between the phosphate group. Okay, that is incorrect. That is incorrect. Held together by the hydrogen bond between the nitrogen, right? Nitrogen base. And the complementary strands are antiparallel and run in opposite direction. That's correct. Okay, so the ones that are not correct are A, B. Oh, sorry. I read the first one wrong. So sorry. First one says nucleotides within a single strand are held together by covalent bond. It's correct. This is talking about the you know, the phosphate and the sugar, right? And then the phosphate and the sugar, right? So this is talking about you know, are the nucleotides held together? What's holding them together? Like, what is that? Right? We actually wrote it down earlier, right? That was the covalent bond. Okay, so that is a feature of DNA. Sorry, I just misread it. Okay. So the answer is B and C. Man, I only plan to have like 30 minutes on this thing, but it looks like we are like way over time. But that's okay. This is another application question, okay? Please try it. I won't launch the poll yet, guys, because it's like blocking you guys. Yeah, but you know, take a look, read it. All right. Okay, anyone else? Gonna close the poll in 10 seconds. Okay, so to do this question, we have to analyze it a little bit. A chemical reaction occurs slowly at 37 degrees. When molecule X is added, the reaction speeds up significantly. Okay. So what is molecule X? What have we learned in the whole lecture that can speed up a chemical reaction? Thank you, Roy. Enzyme. That means molecule X is an enzyme, right? Enzyme catalyzes, speeds up reaction by reducing the activation energy, right? It's like the tuition example, right? That I give you, right? With like, it's like scholarship, right? It makes it lower. So now that we have figured out molecule X is an enzyme, and you should know enzyme is a type of protein, then the question is easier. Which of the following is the monomer of a protein? And so protein is made up of amino acid. And you should recognize which one is amino acid, right? That's the amino acid. The answer is A. This is a nucleotide. We have the phosphate, the sugar, and the nitrogen base. That's a nucleotide. Okay, this is some kind of monosaccharide. Okay, you don't have to know which one, but like at least you should know, oh, hexagon, monosaccharide, right? And then this is just some fatty acids, right? Okay, so the answer is A. Any questions? All right. Now I wanted to start on lecture 3, but I don't know, like judging from the results from the poll, I think maybe you guys could use a review, like a quick review of the whole lecture to prepare you for the quiz. Is that something you guys want to do? Huh? Let me do a poll, okay? If you want to review, choose A. If you think we should move on, choose B. You guys answer really quickly, huh? Compared to the to the poll questions. Very good. Okay, I guess most of you do want to review, so I will go ahead and do that. But let me get some more tea first, okay? Like I am getting dehydrated here. Okay, so I'll be right back. Now I want you to know that there are actually tutorial videos that are available on East Centennial. I don't know if you guys are aware. So let's say you go into the course shell, right? And I would say like usually I would post something like the study guide answers are now available. So if you click on that, you will see the answer, which is just the Word document with the filled in. And then there is also these tutorial videos, right? So these are like old recordings where I took up the study guide. Okay, so like if you want to watch that as a review, as a way to review, you know, it would be a good idea. Now, the quiz is on Thursday, like I said, and you still have to come to class, right? I told you that several times already. So make sure you plan out your day accordingly, okay? Because I know you also have math and stuff, right? Okay, so let's do a quick review for lecture 2. I'm just gonna write down the main concept. These are not the only thing you have to know, but this would be a starting point.
Sorry, Professor, I have a question. You said come to class, you mean online?
Yeah,
Or-
online.
Oh,
yeah.
okay.
So do we still have our virtual session on Thursday, 8:30 to 10:20? So plan to do the quiz sometime during the day.
Okay, no problem. Sorry, I thought you meant go on campus.
No. Yeah, that's okay. Thank you for clarifying. Yeah. Okay, so we are talking about the sorry, macromolecules, right? So the first one is carbohydrates. Okay, so I guess what I'm gonna do is I'm gonna write down the monomer, right? Monomers. Okay, so the monomers for carbohydrates are monosaccharide. Okay, now you should make your own summary sheet, right? You should write a copy along. Okay, this could be your cheat sheet, right, for the quiz. Okay, so for carbohydrates, it's made of C, H, and O. All right, C, H, and O. And again, for those of you who haven't heard this for some reason, right, you are allowed to use your notes for the quiz. So the monomers is made of monosaccharides, and we have 3 separate ones, right? Okay, we have the glucose, we have the galactose, and we have the fructose, right? And so these are the monosaccharides, the single sugar. Okay, and they could come together, they could connect, right? And then the next level up would be the disaccharide. Disaccharide, right? Which, I mean, you can write out the formula, but let's do it. Okay, that's fine. So glucose plus glucose You should know by now, gives you maltose. Gives you lactose. Gives you sucrose, right? So these are disaccharide. And then we have the polysaccharide, right? I mean, there are different kinds of polysaccharides, but then the ones we've done in class, they are just made up of glucose, right? Okay, or in the case of chitin, is modified glucose. So help me out if you would. What are the 4 different kinds that we did? Just type them out or yell them out. Give me some. Nothing? Oh boy. Okay, that's fine. So polysaccharide would be like things like cellulose, right? Okay, yeah. Starch, very good, thank you. Is it
Glycogen.
Lorelli? Yeah, okay. Glycogen, very good. And then chitin, right? Okay, so these are okay. Ramon, insulin is protein, okay, not carbohydrates. Okay, so please remember that. So these are for structure, right? And these are for energy, right? Storage. Okay, so cellulose is fibers in plants, right? Cell wall of plant cells. Chitin is exoskeleton of insects, right? Insects. And crustacean, right? Crustacean, which are like crabs and lobsters and stuff like that. Did I spell it right? Crustacean. C-R-U-S-T-A. Missing A. Crustacean. Starch is storage form. Storage form of energy in plants. You will find them in roots or stem. And glycogen is storage form of energy in animals. Remind me where you find glycogen in the body? 2 places. Thank you, Madison. Liver and muscle. Very good. I will post this afterwards, of course, okay? But try to copy as much as you can. That's pretty much it for carbohydrates. Okay, I mean, those are the key thing to remember. And of course, there are like little things, right? Like food sources, but that's simple, right? You can get it from rice, potato, candies, dairy products, right? All right, next one that we're gonna do is lipids. Lipids. Lipids, we have C, H, and O, and then also P in phospholipid. Okay. Key things to know about lipid is that they are all hydrophobic, right? And components. Components include glycerol, fatty acids, which could be saturated or unsaturated. You might have a phosphate group. And then carbon rings, right? Okay, so they're a little bit different compared to the other three because they don't have monomers. Instead, they have these components, and you kind of put them together to build what you want. Okay. So let's start with the first kind of lipid. Just gotta put one here. Triglyceride, aka fat. Okay. We use this for energy storage, long term though, right? Okay. Which part? Like this last part here, Ramon? Okay. Like I said, I'll post this online afterwards as well. So, you know, whatever you missed, it will be there. All right. So energy storage, we'll say long term. Like when I say long term, I don't mean like years, right? Like I mean like if you're not eating for like a few days, then you start using the fats, right? Yeah, that's long term. Insulation, remember? Insulation of organs, keeping them warm. That's triglyceride. Okay, let's take a look at the components. It is glycerol, 1 glycerol plus 3 fatty acids. All right. And I just want to remind you, all these things are connected through dehydration synthesis, right? Dehydration synthesis. Take in an OH from one, take in an H from the other, pull the water out, and then you form a covalent bond, right? A covalent bond is a bond where they are sharing electrons. So that's 1 glycerol plus 3 fatty acids. Okay, now we also talk about unsaturated versus saturated, right? Fat. Okay, so let's take a look. In terms of the structure, for unsaturated, it is 1 glycerol plus at least 1 unsaturated fatty acid. Okay, and over here is 1 glycerol plus 3 saturated fatty acid. Okay. Now, what is the meaning of saturated and unsaturated fatty acid? Okay, so unsaturated fatty acid means the fatty acid contains double bond, right? Not connected to maximum number of hydrogen. You still have more room for it. It's not saturated yet. Can I repeat that, please? Repeat what? Repeat this last part here. Okay. So the fats, right, is 1 glycerol. Let me show you a picture. The fats is 1 glycerol with 3 fatty acids, right? The fatty acids can be saturated fatty acid or unsaturated fatty acid. Okay, so unsaturated fatty acid means it has a double bond to it. That's all. Right? It has a double bond, and when it has a double bond, that means it's not connected to as many hydrogen as possible. There are still room for hydrogen. That's basically what it means. On the other hand, if it's saturated, then fatty acid has no double bond. No double bond. Okay. They are connected to max number of hydrogen. No more room for hydrogen. Okay. No more room for hydrogen. And this one still have room. Still have room for hydrogen. Okay. No kink. No kinks. And then this one has some, excuse me, some kinks. Someone had a question. I heard someone mic unmute.
Yes, sir.
Yeah.
We do have a quest. We do have a test quiz tomorrow, right?
Thursday.
Thursday.
Yeah. Thursday.
Is it-
It'll be available from 8:30 to 11:59 p.m. Okay.
Okay, and that's the test, right? Our quiz.
Just a quiz. Just a quiz.
Okay.
10%. Yeah. Okay. All right. So that's that. Other things to kind of know. This is solid. Solid at, oh no, sorry, this is unsaturated, right? Unsaturated, so this is liquid. Liquid at room temperature, and this is solid at room temperature, right? I know some of you are thinking, hey, but this is already in my notes, right? Okay. But writing it out is going to help you memorize it. Okay, writing it out is going to help you remember. When you study biology, you should not be reading the notes, okay? Reading the notes is not going to help you remember it. Okay, you have to write it out. Right? That's my advice. Okay, write it out in your own words, right? Do what I'm doing here. Okay, like I actually don't have notes on the side. I'm not like copying over, right? I'm trying to do it from memory, and then you write it all out. Okay, and whatever you don't remember, you go back in your notes and you look it up, and then you add it on with a different color, right? So like those are the things that you missed the first time. Right. So then when you study again on another day, you start again with another blank piece of paper, and then you start writing all the things. And hopefully, the amount of extra things you have to add every time is gonna decrease, right? Because you can recall more and more and more. All right. That's my advice. Okay. The only thing to be careful of is like, just because it's an online course, right? Don't assume you can just look it up all the time. Okay, because time is going to be tight on the quiz. All right, that's what most students, the feedback they told me. Okay, like, you thought you have a lot of time, right? But in fact, not quite. All right, so that's that. That's triglyceride. Another thing. Oh, sorry, I forgot. What if you want to convert one into the other? What if you want to take unsaturated fat and change into saturated fat? What's the process called? Right? Hopefully you're thinking of hydrogenation, right? Okay. Artificially adding hydrogen onto it. Okay, so that's triglyceride. We also have phospholipid. Phospholipid. So this is major component of cell membrane. Okay. So it has a glycerol. It has a polar head, right? The phosphate. I also write hydrophilic phosphate head or group, doesn't matter what you want to call it. And then we have the tail, right? fatty acids. Okay, could be saturated or unsaturated. It doesn't really matter. Okay. That's all we've learned for phospholipids. Okay. I mean, I had a few pictures that shows you how they are arranged in the cell membrane, but you don't really have to know that. Okay, just know that it's a special molecule because it has two different parts to it, right? It has a hydrophilic part, and then it has the hydrophobic tail. Okay, so it's CHO and then a little bit of phosphate as well. There is wax. No questions on wax for the quiz. Okay, and then there's steroids. Okay, you would know it's a steroids because it has 4 fused carbon rings plus some functional group, right? You don't have to know what the functional groups are called. They're just like add-ons, right? That you put on the rings and it gives them special properties. All right. That's that. In terms of functions, what do we need steroids for? They are also part of the cell membrane. Okay. We have the cholesterol as an example of a steroid. And we need the cholesterol to make different things, right? Vitamin D, I think that's one of them in the notes. We need cholesterol to make the biosalt, right, to help break down fat, and you know, hormones. Right? Not necessarily sex hormone. Sex hormone is just an example of it, right? So like testosterone, estrogen, progesterone. Zainab said cortisol. Yep, that is that's actually not an example of a functional group, but that's an example of a steroid, though. Okay, you make that from your adrenal gland. You learn that next semester. That's it for lipids. Okay. I know, right? Like I always say, that's it, but then it's actually a lot of stuff. I do understand that. And proteins. Next one is proteins. Okay, proteins has CHON and trace of what? Anybody remember? Trace of. Okay, medicine said P. Not quite. P is in DNA and a little bit in fat. What is the thing here? Okay, S. All right. Let's see, I'm just gonna go back to the notes and show you that I'm not lying. Okay, right here, trace of S. Okay, because one of the amino acids has sulfur in it, right? Okay, so this one is the monomer again. Monomer. It's 20 amino acids. 20 amino acids. 9 of them are essential, which means you have to eat them. You cannot make it. You don't have to memorize what they are. Just, you know, be aware that some you cannot make, some you need to eat. Okay, so we talk about the structure of the amino acid, right? Like you don't have to draw it, but you should recognize it when you see one. Okay, carboxyl group, amino group, right? Okay, I'm give you a moment to catch up, and yeah, ask questions. I don't know what you don't know, right? I don't know what you need help with. Ask.
Sir,
Mhm.
the diagram that you just drew, that's an
Amino acid.
example of an amino. Okay,
That's right. Yeah.
okay. So and oh,
That
so
is the
of
side
one,
chain.
right?
Of
Like
one amino acid. That's
one amino acid. Okay, so it's like 20 of these equal the protein.
right. I
Or
mean,
no?
you have 20 different options of these, but like which one you put together is determined by your DNA, right? So like you don't have to have all 20 of them, right? And it doesn't have to be 20 amino acid long. For example, like this protein that I showed you in class, like this one, the hemoglobin. This one has 146, right?
Oh,
So like it's not necessarily all 20 together, right? It's mix and match, right? Think of it as like building a necklace, right? With the beads, you can have
okay.
many. You can have a few, right?
Okay, okay.
All right.
But do we
question.
have to like recognize the amino acid, how it looks like? Is that like important?
You don't have to recognize what each of the 20 looks like, no, but you should recognize this is an amino acid. For example, like that last question we did, right? Over here, right? You should recognize that, oh, that's the general structure of amino acid, right?
Yes, okay.
Because, yeah,
Okay, perfect. Thank you.
okay. Great question. Anyone else? Okay. So, where was I? So that's that. And then you should review the level of structure, right? 4 levels of structure. Okay, I feel like we just did it today though, right? Like at the beginning of class. So I don't wanna write it out again, but it's the primary, secondary, tertiary, and quaternary. And it's basically this table, right? Question number 17. Okay, so you should know what each level represents, and you should know what's holding them together. Okay, I'm gonna copy it here. And I just want to remind you, like these 3 levels, secondary, tertiary, quaternary, right, can easily be affected by temperature, pH, salt concentration, right? Okay. But then the peptide bond needs hydrolysis to be broken down. Okay, this is question number 17 from study guide. Okay, so like if you just heat up your stuff, you're not gonna break down the whole protein. Like, I mean, you will denature it. That's the term we use, right? You will unravel it, but then you will still have the primary structure intact.
Sir, can you give an example, like for example, like in the kitchen, can you give an example of the primary? Like the
Can I give you an example of a primary structure?
yes, what you were just talking about right now.
I mean,
Yeah.
all proteins have primary structure though, right? Okay,
I
because
meant like when you were saying something about heating and you heat this and
okay, yeah. So like for example, right, like the egg, right? The egg white. When you cook the egg white turns into the white color, right? So in that process, the proteins that's in the egg white, you basically denatured its tertiary, secondary structure, and then it just becomes a single strand now, right? So this is essentially what happened. Let's say these are 2 different proteins, right? These 2 different proteins, and they're on their own, like in a ball shape, whatever, right? Okay, so you heat them up, you basically unfold it, and it becomes the strand. They still held together by the peptide bond, but now they're just these strands, and then they kind of get tangled up, right, together, and that's what change it into the white color, essentially. I don't know if that helps.
yeah, because I think like when you give these examples of like day-to-day stuff like that we use and do, I think that makes more sense than what
Right.
you know what I mean? Like,
Yeah.
well, that's for me. I don't know for everybody else, but yeah.
Okay, yeah. Thank you for the feedback. I will try to incorporate that whenever I can. All right.
Thank you.
So that is the protein. All right. We did some examples of proteins, right? Functions. We say functions. Okay. Functions. Structure. All right, so like hair, nails, horns, right? And then we have hormones. Example, insulin. I think someone mentioned that earlier, right? Remember, hormones can be lipid hormones. In that case, they are steroids, like the testosterone, progesterone. But they could also be protein hormones, like insulin. Okay, there are a lot of different hormones in the body. You will learn that next semester. And then enzymes. Enzyme speeds up chemical reaction, right? Okay, that's it. That's it. I mean, there was also the picture of an enzyme. I honestly don't remember if I put some questions on it, but the key thing is this. Okay, this is the enzyme, and then this would be like where you fit the molecule in, right? Okay, so this is the substrate. That's the ingredient of the reaction, and then the slot that you fit them in. That's called the active site. All right, active site.
I have a quick question.
Yes, please.
So for the self quizzes, the macromolecules self quiz would be the best one to get us ready for the quiz on Thursday, right?
Okay, so again, the self quiz is a good start, but the actual quiz would not be the same level of difficulty as the self quiz. Okay, the actual quiz would be harder. All right.
Okay.
So like I said, it's a good starting point, but don't stop if you get perfect. You still have to, go through the study guides and stuff like that. All right. Okay. That is the protein, and then we did the nucleic acid, right? Right. I mean, we just did it today. I don't think I should spend more time writing it out. You can definitely do that on your own. Write down the you know what, whatever. Let's just do it, okay? So let's just do it. Why not? I can't start the new lecture anyways, right? We only have like what, 20 minutes or something, right? So nucleotides, sorry, I am losing my mind here. Okay, nucleic acid. Right, so CHO, and what else? Two more things.
PS.
Thank you, P and N. All right. So let's compare the DNA and the RNA. All right, compare the DNA and the RNA. I probably have a chart like this in the study guide, but let's compare it. Okay, so the monomer. What makes up DNA and what makes up RNA? Well, they are both nucleotide, right? Nucleotides. Okay, but this is ACGT, whereas this is ACGU. Uracil. All right. And then sugar. This is deoxyribose. And this is ribose. Okay. Structure. Structure would be double helix, double-stranded, single strand. Okay.
So a helix and a strand is the same thing, right?
Yeah, helix just means it's twisted together, right?
Okay.
Like a spiral, yeah. Yeah, that's pretty much the key differences, right? Okay. So obviously you also have to know your purines, your pyrimidine. If you have trouble memorizing them, put them on the sheet. And the calculation. Right? Let's just do another one, right? Because it's so fun. Let's do 35% G. Okay, I'll give you a moment. Figure out everything else. Okay, figure everything else. Okay, that's correct. Thank you. Is it Nap? Anyone else? Right. So hopefully you're like, okay, G and C goes together, 35, 35. Together they make up 70. Right. So that means the remaining is 30%, right? So 30% A and T together, which means it's 15A and 15T. Okay. Yeah, that's pretty much it. All right. Of course, there's a lot of like other tiny details you have to remember or write down, but that's like this is a good start, I would say. Okay. If you have no idea how to start your study, okay, remembering all these things would be a good start. Okay. And then, do more from there. Okay, since I like you guys, I will tell you the 2 written questions. Okay, remember you have 2 questions where you have to type out the answers, right? Okay, I'll give you an idea of what they are. Okay, so both of them are from lecture 1. Okay, so one of the questions, I will show you a picture. Yeah, I won't tell you what picture it is, but I'll show you a picture, and you will have to describe it using qualitative observations. That's one of the questions. Okay. Nina asked, if we're only given G, should we assume it's DNA? Oh, interesting. Yeah, that's a great question. Okay, so like for something like this, the question would have to tell you is DNA. Okay, you won't be able to do this if this is RNA. Okay, because the pairing only works if it's DNA, right? Okay, but that's a great question.
Sir,
Yeah.
we don't have to send in a rough work, right? Just the answers and submit, that's it.
You don't have to, what is it again? Sorry?
We won't need a rough work to send in after.
No, this is not like math, right? Okay, like in math, I don't know what you guys do, but like you might have to show your work and then take a picture or something. I don't know. But there's no rough work in biology, right? Like you just type the answer, right? Okay. Do you guys okay? You guys with me? Did you guys hear what I said about the long
You
answer
said
question?
one of the question is going to be on qualitative reasoning.
That's right. You write it down, okay? Don't say I didn't tell you, huh? Okay.
And what's the other one?
Oh yeah, good. Of course, right. I'm gonna tell you the other one. But before that, I also want to mention something. I'll also write this out later. But when you answer the long answer questions, it has to be your own words, right? You can't just like copy and paste from. Well, I mean, you cannot copy and paste anyways, like because you'll be using a lockdown browser, but you can't just copy it from the notes. Okay, like it has to be your own words. You have to do some kind of analysis, and you're like, well,
You could use your notes.
Yes, for the
Yes.
100th times you can
Yes,
use your
yes,
notes.
yes,
I told
yes.
you already, right, Georgia? Remember?
Just trying to make sure.
Yeah, you can use your notes, okay? But like you gotta use your own words. It has to be appear that you're explaining it, from your own words, okay? Otherwise, I reserve the right to not give you full marks. Okay, the second question, right? What's the second question? Second question, I'm gonna give you a scenario, okay? And then I'm gonna say you will be running an experiment. And then I will ask you different aspect for the experiment. right? Like I will ask you what's the dependent variable. I ask you what's the independent variable. I will ask you what are the standardized variables. Okay, so that's the second question. We've done plenty of practice for those things, right? So are we all clear? I have given you everything I can, right, to allow you to do the best that you can. So hopefully everybody will get good grades, and you'll be happy, I'll be happy, everybody's happy. Okay.
About how long until we would get the quiz back?
I think probably next day, okay? Next day,
Oh,
if not next
is
day,
that-
Friday for sure. Because I have to mark them, right, for the written
Yeah.
response. Yeah. But Thursday I have like full day of classes from 8:30 all the way to like, very late. But I'll try to get them in, right? But definitely before Friday. Okay. Now, I mentioned this last time. You don't get to see your quiz though, right? You only get the final grades, right? Okay, that's part of how online learning works. You know what? I think I'm gonna leave it at that. I was going to start lecture 3, but I don't think anything's gonna go in. Okay, we only have 20 minutes anyways, and then I didn't give you a break anyway. So that's it. Okay. Come to class on Thursday. On Thursday. Come to class on Thursday. We will do lecture 3, and then you also have to take the time to do your quiz as well. Okay. Remember to find a quiet place that you will not be disturbed. Is this the first quiz you guys are doing for the program?
Yeah.
Yes. Okay. So make sure you. I was looking at the lockdown browser thing. I don't know. Like a lot of you still haven't tried it out. You need to do that. Okay. You need to install your lockdown browser. Otherwise, like if you're doing it the first time on Thursday, things could go wrong, right? Okay, that's all I'm gonna say. If you have questions, feel free to stay behind. Otherwise, email me whenever. And thank you. I'll see you all on Thursday. Okay, bye-bye.